Hello and welcome to this presentation. The title is Monitoring and Assessment of Valued Ecosystem Components in a Constructed Juncus-dominated Salt Marsh on the Northern Gulf of Mexico. This research was largely conducted by our graduate students Nicholas Murphy and Amelia Marshall at the University of Southern Mississippi. A valued ecosystem component is an element of the environment that has scientific, economic, social, or cultural significance. VECs could include air, water, soil, vegetation, fauna and flora, etc. that may be affected by a project activity and are often included in the environmental assessment. Salt marsh VECs can include both the structural and functional elements of the habitat. These could include the sediment properties, the vegetation diversity and biomass, microbial and faunal co-inhabitants, and so forth, and we shall explore a number of these in this presentation. Beneficial use is the practice of repurposing sediments for the purpose of creating additional new habitat. It is often employed in salt marsh restoration or augmentation projects where sediment are used to create elevations that allow salt marsh vegetation to establish and thrive. The Army Corps of Engineers and the Mississippi Department of Marine Resources have used the shores of Deer Island, Mississippi as an example of restoration with beneficial use material. The BU program in Mississippi was mandated in 2010 as an opportunity to increase habitat area. The location of Deer Island, Mississippi is along the northern Gulf Coast, offshore of the cities of Biloxi and Ocean Springs. This remnant spit of land is not a true barrier island, however, it is disconnected from the current shoreline. You can see in the northeastern corner, there are two areas which have been used for restoration using BU. These include a site that was begun between 10 and 15 years ago, and a newer site that was begun between 2 and 5 years ago, and the adjacent reference natural marsh. The figures in the bottom show the youngest restoration site on the left, the intermediate age restoration site in the middle, and the natural marsh on the right. As I said, two beneficial use sites, DIMR1 and DIMR2, have been planted since initial construction around 2005, with a number of native salt marsh and sandburn vegetation species, with the ultimate goal of restoring Juncus roemerianus black needle rush dominated marsh. Black needle rush is the dominant marsh species along the northern Gulf, and this rush is slightly different in both its morphology and density to the more typically studied Spartina alterniflora marshes elsewhere in the country. Observations of the two projects indicate both natural recruitment and establishment of the planted species, with the exception of black needle rush that tended to fail after planting and has recruited poorly. On to the methods. We essentially looked at a number of different VECs, including the sediment microbial diversity, the soil conditions, the elevation of the soil, the vascular plant diversity, as well as biomass. This was sampled in multiple seasons from spring 2017 to fall 2019. Simultaneously, benthic invertebrates and fish use were sampled using both minnow traps and quadrat sampling to enumerate primarily the gastropods and the crustaceans, which focused mainly on fiddler crab use. Finally, sea level rise was projecting using the U.S. Army Corps of Engineers Biloxi, Mississippi sea level rise projection curves for the low, mid, and high scenarios. Here we can see results from the microbial diversity sampling in fall 2016. And I want to draw your attention to the difference in the two plant species, with Juncus falling more along the upper part of this MDS plot and Spartina falling more along the bottom half of the plot. Additionally, you can see that there is a trajectory with DIMR2 in blue, DIMR1 in green, and the pink natural marsh showing a trajectory over this space-time plot. Here we can see diversity of the three different sites in terms of the different microbial groups present, In particular, the gamma proteobacteria, the bacteroidea, and the delta proteobacteria are quite abundant in all samples. The difference between Juncus and Spartina in the natural marsh and DIMR1 is somewhat less evident, although in the DIMR1 site, the Juncus on the right is showing different diversity than the Spartina on the left. The soil organic content was significantly higher in all seasons at the reference marsh compared to the two restored sites. The vegetation species richness using both quadrat-based and point intercept-based sampling shows that the intermediate age site was the site with the highest species diversity. This was largely because the natural marsh is dominated by a mix of Spartina alterniflora and black needle rush with very few other species present. Looking at the plant biomass results on the right of this slide, you can see that in all seasons, the below ground biomass fraction was significantly higher than the above and live and dead fractions. And this was the case in all seasons, and the highest biomass was found in the natural reference marsh. Similarly, on the bottom, you can see that between 50 and 85% of the total vegetation biomass collected in the core was found in the below ground fraction in the reference site, and this dropped to 50% or slightly higher in the two restored sites. Given this together, the two restored sites did not yet have similar soil organic content or below ground biomass as the natural reference condition. Here we can look at the nitrogen and carbon content of the sediment cores on the left and the three tissue fractions of the plants on the right. And we can see that the natural marsh had significantly higher sediment nitrogen and carbon, and the above live fraction was the portion that had the highest amount of nitrogen in the plant tissues. There were some differences among the two species, Juncus on top, Spartina on the bottom. Primarily, the amount of nitrogen was higher in the above live tissues of Spartina than it was in Juncus. Carbon was slightly greater in the below ground tissues of Juncus than it was in Spartina. The third VEC are the benthic invertebrates, the primary consumers of the vegetation and detritus. Here we can see 4 different species: fiddler crabs enumerated by the number of burrows, ribbed mussels, periwinkles, and olive snails. In all 4 species, the abundance was highest in the natural marsh site on the right of the plot, with both ribbed mussels and periwinkles being conspicuously absent in both of the 2 restored sites. This feeds up to higher trophic level consumers, in particular the clapper rails are known to be heavily dependent on fiddler crabs during the nesting season for a fat-rich food source, and we can see that again the abundance of clapper rails mirrors the distribution of the fiddler crab burrows, with the highest abundance of clapper rails in the natural site, slightly higher in the older restored site than the younger restored site. Seaside sparrows also more abundant in the 2 older sites than the younger site. Finally, we'll look at sea level rise, and we can see here the lowest and highest sea level rise curves for the 3 different site elevations. If we map these out in 2050 and 2075, the blue areas indicate areas that are inundated, showing that the majority of sites have a future lifespan of between 50 years to 70 years. The conclusions are that the constructed sites do have higher and greater elevations, with a number of implications on both salt marsh diversity and other VECs, and that the natural site in particular tended to support more valuable VECs than the 2 restored site, although this may be a function of age, and we expect some of these parameters to converge as the sites continue to age. Finally, the sites do have a finite time horizon, with complete inundation occurring between 2070 and 2100, especially at the highest sea level rise scenario. Other VECs of importance could include slash pine forest, the ecosystem services of storm reduction, recreation, and fisheries.